Assalamualaikum and good morning. Today, we from AS2572E, we're going to present about Saccharomyces boulardii. First of all, I'm going to introduce our group member. The first one is Damia Adawiyah binti Mama Ali. The second one, it's me, Nur Akmal Madrisha binti Mama Anwar. The third one, Anu Shazwa Fatira binti Rizbi. The fourth, Shira Nur Ashiki binti Sofan. And Nurul Farahia binti Muhammad Fakrul Amin. And the last one is Nur Rashida binti Rozani. For introduction, Saccharomyces boulardii is a type of probiotic which is friendly organism. It's a yeast that is actually a strain of Saccharomyces cerevisiae. Friendly organisms such as Saccharomyces boulardii can help to fight off bad organisms that might cause a disease. Saccharomyces boulardii is different from other strains of Saccharomyces cerevisiae, which commonly known as brewer's yeast or baker's yeast. People commonly use Saccharomyces boulardii to treat and prevent diarrhea, including rotavirus diarrhea in children. It's also used for other types of diarrhea, acne, a digestive tract infection that can lead to ulcer and many other conditions, but there is no good scientific evidence to support many of its other use. Next, we move to kingdom and binomial nomenclature. First of all, we start with kingdom for Saccharomyces boulardii, which is fungi. Phylum, for phylum, Ascomycota. Class, Saccharomyces. And order, Saccharomyces. For family, Saccharomyces. And next, genus, Saccharomyces. And the last one is species, which is Saccharomyces boulardii.
We move to the characteristic of the microbes. Morphological of Saccharomyces boulardii, they have cell wall. Their cell wall is made of chitin. The function of chitin is to make the cell wall rigid and thick. And they also have a mitochondria, vacuole, plasma membrane, nucleus, endoplasmic reticulum, Golgi apparatus, cytoplasm, and ribosome. They also have a free ribosome too. Look at the dot. That is a free ribosome. Nutritional for Saccharomyces boulardii is chemoorganotrophic and heterotrophic. The carbon source of Saccharomyces boulardii is simple sugars like sucrose, fructose, and glucose. The energy source is organic compounds, and the nitrogen source is ammonium salts, amino acids, and peptides. For vitamins, J requires beta-complex vitamins like biotin and thiamine. For minerals, J requires phosphorus, potassium, magnesium, sulfur, and iron. For oxygen requirement, they need facultative and aerobic. They use aerobic respiration when oxygen is present. And also, they use fermentation when oxygen is limited. For the growth medium, they grow well on sorboroid dextrose, agar, and other sugar-rich media. For pH references, they are slightly acidic, like pH from 4 till 6 only. And then temperature for optimal growth around 30 to 37 derajat Celsius.
Now, let's take a look at the habitat of Saccharomyces boulardii, which is a tropical yeast with a unique natural habitat compared to other common yeasts. The primary natural habitat of Saccharomyces boulardii is the skin peels of tropical fruits, specifically lychees or known as Lychee chilensis, and mangosteens that also called as Garcinia mangostana. In these environments, the yeast thrive on the surface of the fruit where it is exposed to high temperatures and varying sugar concentrations. Unlike most Saccharomyces that prefer cooler environments, Saccharomyces boulardii has evolved specific traits to survive its tropical habitat, which are heat tolerant, that it can grow optimally at 37 degrees Celsius, higher than other Saccharomyces cerevisiae that prefer 30 degrees Celsius. It also has high resistance of acid until pH 2.0, which is an adaptation that likely developed to survive the acidic environments of fermenting fruit or through the acidic stomach of an animal that consumed the fruit. Lastly, it has nutrient competition. Saccharomyces boulardii produce high levels of acetic acid that helps to inhibit the growth of competing bacteria in its natural environment. Next, we take a look at its reproduction. In typical Saccharomyces yeast, sexual reproduction occurs through sporulation, but Saccharomyces boulardii has a significant reproductive quirk that it is sporulation deficient. In nature, this means it almost exclusively relies on asexual cloning rather than sexual reproduction. It is because research has shown that Saccharomyces boulardii is diploid that has two sets of chromosomes, but it also has lost the ability to sporulate effectively. Scientists believe this deficiency might be an adaptation to its stable tropical habitat. Saccharomyces boulardii reproduce primarily through asexual budding, which is a process that a small daughter cell grows from a mother cell and will eventually detaches. Firstly, the small protrusion will form on the mother cell wall. The DNA of mother cell replicated and the copy of that DNA was transferred into the growing bud. Saccharomyces boulardii can double its population between 90 to 120 minutes with an optimal conditions. At the end, a new, genetically identical yeast cell is formed and a single mother can produce about 20 to 30 buds in their entire lifetime before reaches replicative senescence, which is also known as old age.
Next, I will talk about the significance of Saccharomyces boulardii. The first one is a probiotic yeast. Saccharomyces boulardii is a non-pathogenic yeast which not causes a disease and considered as a probiotic that widely used in the medicine and health industry. The second one is support gut barrier. This non-pathogenic yeast has some useful properties such as survive in acidic environment and enhance the production of secretory IgA that play crucial role in protecting the mucosal surface of the gastrointestinal tract and helps maintain a healthy gut environment. The next one is treatment for ADD. In case of ADD, antibiotic use disrupt the balance of bacteria in the gut, which allowing the harmful bacteria to thrive. Saccharomyces boulardii is effective in treating ADD because it is resistant to antibiotic and has the ability to restore the gut health. Next is protect against a pathogen. Pathogen that produce toxins such as E. coli and Salmonella will bind to this probiotic and inhibits harmful pathogens. Therefore, it will reducing the risk of gastrointestinal infection and neutralize the toxin from the pathogen.
Now, I would like to explain more on the significances of Saccharomyces boulardii. First, Saccharomyces boulardii inhibits the activation of nuclear factor kappa B or NFKB, the encode cytokine and chemokine, the regulator of inflammatory responses. Saccharomyces boulardii produce a factor that blocks the signaling pathway that produces cytokine and chemokine. This can prevent the overstimulation of inflammatory responses. For example, Crohn's disease, the disease due to inflammation in the gut due to overstimulating of inflammatory responses that lead to diarrhea. Next, we move to the second significance of Saccharomyces boulardii, which is Saccharomyces boulardii increase the digestive enzyme production and stimulate a balanced microbiome. It enhances the ability of the gut for metabolism and absorption essential nutrients. What are the enzymes we secreted by Saccharomyces boulardii. They are maltase, sucrase, lactase, 54-KDA protease, and alkaline phosphatase. We all know that maltase, sucrase, and lactase are enzymes to digest disaccharide. So maltase, sucrase, and lactase improve carbohydrate digestion and glucose absorption. This increases energy production, as we all know that glucose is the main substrate for cellular respiration to produce energy in ATP. Next, we move to 54-KDA protease. 54-KDA protease is not a normal protease, not a common protease. It detoxifies toxins A and B from Clostridium difficile that cause a symptomatic gastrointestinal tract infection. Next, we move to the last enzyme, which is the alkaline phosphatase. As its name, it relates with the phosphate group. Alkaline phosphatase inactivates the E. coli by removing the phosphate group from the lipopolysaccharide of E. coli. So it can make that E. coli is no longer toxic to the gut. And last, we move to the last significances of Saccharomyces boulardii. Saccharomyces boulardii promotes the integrity of tight junction of the gut cell. Normally, gut cells must be tightly together. So, what Saccharomyces boulardii do? It promotes the expression of occludin and claudin, the transmembrane protein that are crucial for the formation of tight junction. So, it can make the gut cell be tight together so to prevent from any harm material such as bacteria from pass by from the cell and enter the bloodstream. That's all for me. Thank you.
In this slide, we are focusing on the commercialized products of Saccharomyces boulardii. Saccharomyces boulardii is most commonly probiotic used in pharmaceutical products. Saccharomyces boulardii is predominantly used as a therapeutic agent to prevent and manage gastrointestinal disorder. This probiotic can be effective against antibiotic-associated diarrhea as well as Clostridium difficile infections. In terms of its use, it's effectively reducing the incidence of diarrhea, low the chance of developing traveler diarrhea, and also reductions of Clostridium difficile infections. Examples of the commercialized probiotic products that can be found in the market include Enterol, Florastol, and Atralibium. They are available in capsule or powder form. Next, we take a look at the use of Saccharomyces boulardii in functional food and beverage. Saccharomyces boulardii has been added to a number of functional foods and drinks, particularly in dairy-based products. It offers health benefits including antibacterial, antitoxin, immunological modulation, and trophic action. It has the ability to treat a variety of gastrointestinal and inflammatory disorders. In terms of its use, it improves intestinal microbial stability, support gut health and digestive balance, and function as an effective probiotic culture. Examples of the commercialized dairy product that can be found in the market include yogurt-type probiotic formulation and kefir.